Okay, good morning, everybody. We are on a pretty tight agenda, so let's start, even if some people are still sitting. So as I hope you know, this is the system session. If you're not meant to be here, then you're in the wrong room. And systems is basically everything, right? So we're dealing with lots of data, we're dealing with dynamic models, we're dealing with networks, etc. And then during these 3 sessions that we're going to have in the ECCB 2022, that's one now, one in the afternoon, and one tomorrow in the morning, we will have a little bit of everything. And in this first session, we will see a little bit of metabolic modeling, some cell differentiation induced by a small molecule, and some more modeling and diffusion models based on Boolean models, right? So without more ado, let's start with the first speaker. This is Ryan Toulier from the CNRS in France. So remember, 15 minutes, I'll give you a sign.
Okay. Hello, everyone. I'm very glad to be there this morning at this wonderful conference to present you our work on how to learn metabolic regulation rules from time series data with our approach, Marine. So, for this work, we consider a cell like an hybrid multi-layer structures composed of 2 interconnected systems. In the first layer, we have metabolic systems, so all the chemical reactions that will be used by the cell to transform substrate into energy and biomass. And for the second layer, we have regulatory systems, which are all the rules that that allow the cell adapting itself to its environment. And to do that, the regulatory system will use signals issued from the metabolism and its substrate, and return controls that will inhibit some chemical reaction of its metabolism. And here in our work, we aim to infer the regulatory system, so this yellow part here, from time series observation of the cell coming both from the metabolism and the regulatory system. One kind of behavior that we aim to explain through the regulatory rules is the diauxic shift. So diauxic shift, it's a complex cell behavior characterized by successive growth phase on different carbon sources. And it is known that it is controlled by regulations. So here we have a small simulation of cell biomass growth on different carbon sources. So it is a diauxic shift. This simulation is divided in 4 phases. In phase A here, we can see that the cell grows on one substrate, carbon 1, until its depletion. Then in phase B, we can see that we have a lag phase, so there is no growth anymore, because some internal regulatory rules prevent the cell to consume carbon 2, so the other source of carbon. And then when the system finishes to update itself in reaction of the depletion of carbon 1 after phase A, we start in phase C, where the cells start to grow by consuming carbon 2 until its depletion. And so here our objective is to learn all the regulatory rules that allow explaining these cell behaviors or those cell behaviors, but and explain why here in phase A carbon 2 is not consumed, why we have this lag phase in phase B. But we cannot just learn any rules that we want. We must restrict ourselves to some kind of biologically feasible rules. And to do that, we need to take into account some prior knowledge. So this knowledge, it takes the form of some specific interaction, either inhibition effect or activation effect, that will be used to define our Boolean regulatory rules. So we model this prior knowledge with the prior knowledge networks. Here is a simple example where we have 3 nodes. So HX here is a metabolite, RPH is a regulatory proteins, and R8A is a reaction. And we have 3 interactions that is allowed. So for instance, for the case of RPH, we have the regulatory rules of RPH can only depend on the activation of HX and the inhibition of R8A. And that means that from these 2 interactions, we can derive 6 potential Boolean regulatory rules. So the case 0 and 1, which is always inhibited, always activated, and 4 other rules that are a combination of HX or not R8A with the and or logical operators. And if we do the same with the two other nodes, we can see that we have 2 rules for hashtags, 3 rules for array. It will define, and from that we know that any regulatory networks will need to match the rule for each one of those nodes, meaning that here there is 36 compatible regulatory networks. And so the big point here is that the search space that will be defined by a prior knowledge network will be doubly exponential by the number of interactions in the prior knowledge network graph. So the problem that we tackle in this study is the following. We aim from a prior knowledge network and a set of time series data to infer the set of all consistent regulatory networks and here by consistent we mean that respect the admissible interaction defined in the PKN, and that allow to reproduce the input time series data. So we know that from the literature there was already some approach that allow learning regulatory rules from time series data and PKN. These approaches are based on constraint programming and rely on a discrete modeling of the regulatory system dynamics. But the big point here is that those approaches can only use observation of the regulatory systems. And thus, we cannot just use metabolic observation to infer regulatory system from them because they do not allow matching the dynamics of the metabolism. Because in fact, we often model both systems with drastically different dynamics. For the regulatory system, we have often a discrete dynamics, so we rely on a set of logical rules, and we update a Boolean vector by just applying or a subset of these rules. And for the metabolic system, we often rely on a steady-state approximation of the system. So, for instance, we can use a dynamic flux balance analysis to do that. So we will solve successively different linear programming problems to simulate the cells, the metabolism of the cell. And this is this part that is not considered with other approach of the state-of-the-art to learn metabolic regulatory rules. So here we present our approach, mirroring, that allow from a metabolic network, a prior knowledge network, and time series data coming both from the regulatory system and the metabolic systems to infer all the regulatory networks that are consistent with both the backend and the time series, so both with the regulatory dynamics and metabolic dynamics. To do that, we rely on simulation formalism that mix both the regulation and metabolism system, which is the regulatory flux balance analysis. So the idea behind RFBA is to divide the simulation into time steps, and for each time step, just iterate over 3 steps. First, updating the regulatory system, so we apply or just a subset of our Boolean rules to update our states. It gives us a new set of inhibited chemical reactions. Then we update the metabolic systems, so we solve a linear programming problem, and we fix to 0 all the inhibited reactions so that they do not participate in the simulation. And then we finally update the cell environment by adding and removing the consumed or produced substrates. So this formalism has been implemented directly in our inferring workflow. So to do that, we rely on 2 solvers. So we have a hybrid solving approach. So we rely on 2 solvers, one logical solver and ASP1, and a linear solver. And the idea here is that the first solver will find candidates rules according to the discrete dynamics of the regulatory systems and a discrete abstraction of the metabolism, and then send candidates to the linear solver that will solve a regulatory flux balance analysis simulation to check if it matches the observation, and then just accept or reject the solution according to if it matches the observation. And during the reject phase, we add new constraints dynamically to the ASP solver. So these constraints are based on monotonic properties that allow on the regulatory network structures, and these properties allow accelerating the solving process by pre-filtering the wrong solutions thanks to another generalization of counterexample. And so this workflow has been validated using a gold standard instance provided by the literatures. The instance is based on E. coli. It contains 20 reactions, so all these black arrows, 4 regulatory proteins, so these 4 nodes, and 11 regulations, so all the colored arrows, and can model complex behaviors such as the diagnostic chips that I showed you just before. So from this network, we can define its influence graph. So all the interaction between the component, its regulatory system. So for instance, here we have an inhibition effect because there is an inhibition between RPCL and TC2. And thus, to validate this, the approach on this model, we need to define our source space and define our instances. So to define our source space, thanks to the PKN, so prior knowledge network, we start from the gold standard influence graph, since this is all the regulations that we aim to retrieve, and we extend it. So we remove the interaction sign. So we don't know anymore if it's RPCL, which has an inhibition effect on TC2 or an activation effect. Then we remove the interaction direction, so we don't know if RPCL impact TC2 or in TC2 impact RPCL, so we have both directions here. And finally, we have some hypothetical age, some as a carbon-2 or TC1. It results in prior knowledge networks that define a source space containing about 3 trillion potential Boolean networks. And so, to complete this instance, we need some time series data. So we use 5 simulated experiments that are provided with the gold standard in the literatures. So we reproduce this experiment using regulatory flux balance simulations with the tool FlexFlux. And these 5 simulations allow reproducing different environments for the cell. So we have different carbon source, different aerobic, anaerobic, etc. And must test almost all the regulation according to the literatures. And so when we apply marine to this instance, we can see one thing first that is great, is that the gold standard is in the set of inter-informed model, so it's a good point. But if we focus on the smallest network for to be more parsimonious, we can see that we infer one smallest Boolean network, which is a subset of the gold standard network. And so by subset, we mean that all the infer regulations are in the gold standard. But we can see that we do not infer all of them. So we can ask us, did we fail inferring missing regulations? In fact, not really, because we can exactly reproduce the input time series by just running a regulatory flux balance analysis of this model to reproduce the input simulation. And we can also explain why some regulation has not been recovered. So for instance, here for arrays, we can see that this regulation never impacts the regulatory flux balance simulation. And so we need more information to be able to capture it, such as the enzyme decay. And thus, here we have found for the validation part a more parsimonious model than the gold standard that allows to explain the same simulations, the same experiments. But we go further to study how the data type impacts the infer elements, the infer regulatory rules. So we study both observation of regulatory and metabolic systems with quantitative and qualitative measurements. So we allow marine to support 3 data types. First, transcriptomics. So we have globally qualitative information about the whole system. We have fluxomics data, so quantitative information, quantitative information, so rate of the metabolic reactions. And kinetics data, which is quantitative observation of the substrate concentration. And marines support any combination of these 3 data types. And so to generate a testing benchmark, we start from our 5 input simulations, and we select 4 data type combination of interest, and add randomly 6 noise rates, so ranging from 0% to 50%. And it results in a benchmark containing 240 instances. So we test the robustness of marine according to the noise and the type on this instance, this benchmark. So we evaluate the infer result with 2 metrics. For simulation reproducibility, we use the residual sum of square, and the regulatory network structure, we use precision and recall. And we find that, in fact, yes, there is some impact of data type and noise on the inferring quality. But we also see that if we have at least transcriptomics and kinetics data in our inputs, and a noise which is less or equal to 20%, then we have an inferred model which matches the subset minimal one that I showed you just before, so which is the parsimonious model on the perfect data. And so here we have a RSS which is strictly less than 1, and precision recall matching the parsimonious model. So to conclude on this work, here we introduce the approach mirroring, which allow inferring metabolic regulatory rules from time series data, provide both from the metabolism and the regulatory rules, regulatory system. This approach relies on hybrid resolution framework, so ASP and LP, and we have validate and test its robustness with a benchmark based on gold standard issued from the literatures. But as we have some future works here, such as, because we do not have test the scalability of the approach to genome scale metabolic networks. So we know that regulatory flux balance analysis scale well to biggest networks, but here the pk should be the issue, size should be the issue. And we are currently trying to work on a Nicoli core medium scale model to test the approach. Furthermore, we aim to study why some regulations have been unrecovered or recovered. So we study the impact of the experiment data, explain which experiments, explain the inferring of which regulation, and the impact of data type and noise on that. So maybe we will work on more precise simulation formalism, such as the RDF-FBA, which allow to consider both enzyme and synthesis in the simulation framework. Thank you for your attention.
Okay, thank you very much. Great talk. Guys, I forgot to say that you can ask questions through the app if you don't want to shout them loud. Questions? Okay, maybe I can start. So how good is your model to, for instance, infer wrong information in the prior networks?
Can you just rephrase the question, please?
Yeah, so so you allow for some flexibility in the prior knowledge that you use for the networks, right? So is there, I mean, do you get signals that are very strong suggesting that the prior knowledge that you're using in principle is not good, and then this allows you to reformulate the initial networks?
Okay, globally there is no issue. We do not infer any false interaction during the inference process. When we have less than 20% of noise, with bigger noise, there is some false interactions that appear or missing ones.
But this is when you're simulating noise. So what I mean is, if you use the real prior knowledge and you use the data that you have, so is there any, have you ever got any hint that the prior knowledge is wrong and then you can sort of correct it?
For instance, we do not have hints because we need to know the solution to check if the inference result is good for the moment. But we know that from the solution, we know that in all this model that is inferred by with the perfect instance, we have some interaction that is inferred sometimes, but that do not impact the quality of the reproduced simulations.
Okay. More? Oh, sorry.
Thank you. Very nice talk. I have a question concerning the input data. You said you have transcriptomics, but it's a qualitative data. So I wonder, what does that mean in practice? Like, it's binary or what?
In practice here, we use just Boolean variable to model the transcriptomics data. So either there is an activation or not, so 0 or 1.
Okay. Have you considered the actual gene expression values, like in the system? Is higher expression or lower expression?
We do not test that for the moment. And I'm not really a specialist for this input. We rely on collaborators, which are biologists, and maybe check with them.
Okay, yeah, thank you.
Okay, last one.
Thanks for the talk. I was wondering, like, what kind of time steps are you using for simulating your model, which is a combined one between a Boolean model and a metabolic one? Because I guess that if you use the same time steps, then perhaps as a simplification, we could have just chosen a Boolean model since, I mean, this is going to depend then in the outputs of your system, which...
Here for the inference process, we do not really have some time step between the simulation, since in fact we'll just learn the regulation for one time step without knowing how long it's been between two time steps. And we just do a flux balance analysis and just inhibit some reactions. So we do not run the whole regulatory flux balance simulation. We just run for one time step and check if it matches the observation for this time step according to this observation. So we learn the regulatory state for a time step and solve it to check if it matches the observation. So we do not use a time step duration during the simulation process, the inferring process.
But then you will have to use like real time series to validate it. So you will have to give like a meaning of the time, right?
Yeah.
Like as an actual time.
I'm not sure to understand.
Like the time steps of the simulation,
Yeah.
will have to, like the results of your simulation will have to
for
be
just the time step, we use the same as the simulation process as input. Okay. So I don't remember how much it was, but we've used the same.
Okay, thank you very much for your talk. We have to save the rest of the questions for the coffee break. And let's move to the second speaker, who is Michio Iwata from the Kyushu Institute of Technology. Please.
Hi. Thank you for the introduction. I'm Michio Iwata, and Associate Professor at the Kyushu Institute of Technology of Japan. Today we will talk about small compound-based direct cell conversion with combinatory optimization of pathway regulations. Recently, regenerative medicine via induced pluripotent stem cells, iPSCs, is popular. First, source cells such as fibroblasts were initialized to iPSCs. Then the iPSCs were transformed to target cells such as neurons by transfecting specific genes responsible for the differentiation. On the other hand, directly programming, DR, can directly convert source cells to target cells without bypassing iPSCs. An advantage of the directly programming is the less time to obtain target cells. However, the gene transfection-based directory programming has a serious problem. Because of the use of virus vectors for the gene transfection, there is a carcinogenic risk of virus-induced gene insertion and mutation. To solve the problem, small compound-based directory programming has been receiving a lot of attention. In this case, there are several advantages. For example, the carcinogenic risk can be avoided because the virus vectors are not used. There are several previous studies that report that directory programming can be induced by small compounds. For example, fibroblasts can be directly converted to neurons by adding these small compounds. In the previous study, the small compounds were experimentally identified, but such experimental identification of candidate compounds is costly and time-consuming. Also, the mechanism of small compound-based directory programming remains unknown. We have three objectives in this study. First, we elucidate the mechanism of small compound-based directory programming. Second, we develop a new computational method, Comprendre, to predict new combinations of small compounds for directory programming. Finally, we predict new combinations of small compounds inducing directory programming from fibroblasts to neurons or cardiomyocytes. First, we explain the elucidation of the mechanism of small compound-based directory programming. In this study, we identified the DR-relevant biological pathways by network analysis of the target proteins of the DR-inducing compounds. First, we obtained the information of combinations of DR-inducing small compounds from the literatures and explore the target proteins of the on the small compounds in using the compound protein interactome data. Finally, we identify the biological pathways of the target proteins as the DR relevant pathways. Here we show the numbers of the detected pathways by the pathway analysis. For the conversions from fibroblasts to neurons, we tested 6 sets of compound combinations and we detected 90 pathways. Also, the conversion from fibroblasts to cardiomyocytes, we tested 7 sets of compound combinations and we detected 102 pathways. There are a number of common pathways between neurons and cardiomyocytes. For the neuron-specific pathways, we detected 3 pathways. For example, Wnt signaling pathway is detected. This pathway is required for basic developmental processes, and it has been reported to be involved in the directory programming of neurons. So the detected pathways are biologically relevant. Also, the cardiomyocyte-specific pathways, we detected several biological pathways. For example, we detected JAK-STAT and PI3K-AKT signaling pathways. These signaling pathways have to be involved in the directory programming of cardiomyocytes. Also, the last signaling pathway is known to regulate PI3K, so the detected pathways are also biologically relevant. For the common pathways, we detected a number of pathways. For example, there are several signaling pathways for cell proliferation and differentiation. So the detected pathways are biologically relevant. So these results suggest that the mechanism of DR-inducing small compounds can be identified by the pathway analysis. Next, we predict new combinations of small compounds for DR. We predict new combinations of small compounds by optimizing DR-relevant pathway regulations. First, we selected some compounds, and for the compounds, we explored the target proteins. To evaluate the functional association of target proteins, we identified biological pathways. We refer to the identified pathways as controlled pathways. If the pathways regulated by selected compounds were similar to DR-relevant pathways, the set of small compounds was predicted to be the candidate combination for DR induction. In this study, toward clinical applications, we focused on the 5,000 approved drugs for candidate compounds. It is challenging to consider the combination of 5,000 approved drugs because of combinatorial explosion problem. For example, if 3 candidate compounds were given, there are 4 combinations, and 5 candidate compounds are given, there are 26 possible combinations. So as the number of candidate compounds increases, the numbers of candidate combinations increases exponentially. Therefore, the combinatorial optimization algorithm is required. We introduced a variant simulating annealing algorithm for the combinatorial optimization problem. Here we illustrate a situation in which the candidate solutions, objective functions has 2 candidate solutions in the searching space. With the simple algorithm, it is very difficult to obtain the optimal solution from the initial guess. In contrast, with simulated annealing algorithm, owing to the deteriorated search for the next solution at the given possibility, the local solutions can escape from the local solution and reaches the optimal solution. We construct the objective function for the combinatorial problem. We define the following equations that consist of three sub-objective functions. The first term evaluates whether DR relevant pathways are covered by the controlled pathways of selected compounds. If controlled pathways include many DR relevant pathways, the score will be higher. The second term evaluates whether DR irrelevant pathways are excluded from the controlled pathways of selected compounds. If controlled pathways exclude DR irrelevant pathways, the score will be higher. The third term evaluates the number of compounds in the predicted combination. Toward clinical applications, the smaller number of compounds are desired because there is a risk of side effect due to the combinations of multiple compounds. If the numbers of compounds is small, the score will be higher. We apply the proposed method. Proposed method to direct cell conversions from fibroblasts to neurons or cardiomyocytes. We search for new combinations of small compounds inducing DR for each direct cell conversion. Here we optimize the objective function using the simulated annealing algorithm. Here we show some examples of the output of the objective values. In the case of the total objective function, the iterative calculation can successfully converge it to the maximal value. Also, in the case of subjective functions, F pathway can show the controlled pathways can include the DR relevant biological pathways. For the case of the subobjective function F known pathway, the controlled pathways tends to exclude the DR irrelevant pathways. Also, in the case of subobjective function F number, the number of compounds in the predicted combination tends to be limited. So the proposed method can effectively identify the optimal combinations of small compounds. Here we show the distribution of prediction scores for the several known combinations. Each box plot represents the distribution of prediction scores for 10 predictions. From this figure, in almost all cases, the new combinations are predicted with high prediction scores. Here we focused on the set numbers 4 and 11. Here we show an example of new compound combination for DR from fibroblasts to neurons. We predicted 2 compounds as shown here. To evaluate the validity of the predicted compounds, we compare the known combinations consisting of 4 compounds, as shown here. The number of compounds in the predicted combination is smaller than that of the known combination. Both combinations include the false scoring, so the rest of compounds could be replaced by this compound in the predicted combination. There is no chemical similarities and commonalities of target proteins. Similarly, we show an example of new compound combinations for DR from fibroblasts to cardiomyocytes. We predicted 4 compounds as shown here. To evaluate the validity of the predicted compounds, we compared the compounds in the known combination. The number of compounds in the predicted combination is smaller than the known combination. Both combinations include the phosphorine and valproic acid, so the rest of compounds could be replaced by these compounds. There is no chemical similarities, but some compounds share the same target proteins. Next, we examine the all prediction results. Here we show the bar plots which explain the numbers of the compounds in known and predicted combinations. The blue bar indicates the number of compounds in known combination, and orange bar indicates the number of compounds in predicted combination. This result suggests that the number of compounds in predicted combinations is smaller than that in known combination. Finally, we examine the compounds that tends to be included in the predicted combinations. Red indicate the known compounds in known combinations, and blue indicate the compound that is functionally similar to known compound. These results suggest that the known and functionally similar compounds tends to be included in the predicted combinations. The red compound is known to be the important factor for various cell conversions in direct reprogramming. So the predicted combinations consisting of red compounds could be a promising candidate combinations for DR. We summarize this talk. First, we identified the mechanism of DR-inducing small compounds at the pathway level. Then we developed a new computational method to predict combinations of DR-inducing small compounds by optimizing DR-relevant pathway regulations. The proposed method is expected to be useful in regenerative medicine. Thank you for your attention.
Thank you very much. Questions?
Hello, nice talk.
Hi.
So in simulated annealing, you have to find a trade-off between exploration and exploitation. And from your graph, you look like you are reducing the temperature slowly and progressively finding this plateau. But there are other schemes. Sometimes you can do, for instance, like waves and going back and, different schemes.
During optimization, yes.
Exactly. Or maybe like spending more time exploring the parameter space and then focusing on something. So I was wondering whether you tried those different alternatives.
Yes, thank you for your question. But in previous surveys, our team, we cannot observe such a situation. So the objective function is fluctuated. So so in practical situation, there could be the problem. So we have to consider the problem. So I have no idea now. Thank you.
Well, thank you for the nice topic. I wonder, many of these small compounds, I guess they target the same proteins.
The same compound?
The same proteins.
Same proteins, yes.
So they are redundant for a target protein.
Mm-hmm.
So how do you, does it make sense maybe to remove redundancy at the beginning of the algorithm to have less combinatorics, for instance?
So we focus on the regulated pathways from detected using the target proteins of the compounds. So at the pathway level, both combinations have the same regulation in the biological system. So our proposed method can perform the well. Sorry.
Yeah, very nice talk indeed. So have you validated your predictions of this mix of compounds that you claim would do DR, or do you plan to, and if so, how?
Yes, thank you for your question. We now planning the in vitro validations with our collaborators. So for the cardiomyocytes or hepatocytes target cells. So we will report the results in the future.
Very last one.
First of all, thanks a lot for the talk. It's very interesting. I have a couple of points, if you don't mind, if you can elaborate about. The first one, when you describe the pathway analysis, can you please describe more about the structural, like the structure of the matrices, how they look like when you extracted them from the pathway? I know it's more technical, but it's interesting. And perhaps if you can recommend some tools actually to extract these matrices from the pathway graphs and how that can actually work. I mean, it's more technical, but it's interesting. There's also a second question.
Yes.
I noticed when you analyzed the pathways, you use more statistical approaches. And is that because it's your own preference, or there is actually a reason you chose it instead of machine learning or deep learning? Yeah, thank you.
Yes, the first, yeah. Yeah, I don't know. I don't understand your question well, but the first question is about pathway analysis. So we use the pathway information from KEGG database. So we perform the statistical evaluation for the pathway enrichment analysis. Then the second question is about the machine learning things. In this study we simply perform the pathway enrichment analysis by the our algorithm, but there is a few rooms to improve the approach using the machine learning or deep learning methods. Thank you for your question.
Okay, thank you very much. And the last speaker of the session is Anastasia Solomatina from the Technical University of Dresden and the MPI for molecular cell biology and genetics.
Yeah, thank you. So, good morning, everyone. My name is Anastasia. First of all, I would like to thank the organizers for this wonderful conference. I'm looking forward to next days. Today, I'll be talking about design centering and how it enables robustness screening of pattern-forming models. So I came to this conference from Dresden, which is in Germany, and there I work at the moment in the Max Planck Institute of Molecular Cell Biology and Genetics. There are many biologists in this building who are working on understanding how cells form tissues. If you have a look at this light sheet microscopy video of a developing embryo, right away we can appreciate how complex and well organized the dynamics of these white structures that are the cells. And during this interaction and dynamics, the functional tissue emerges and eventually a functional organism. And the mechanism that drives these dynamics is poorly understood. Next to Max Planck Institute, there is another building that is a golden building that is called Center for Systems Biology in Dresden, and there computational scientists work also on understanding the mechanism of tissue dynamics, and they use computational tools to understand this mechanism. So actually today, so in biology there are many patterns, and in developmental biology there are even more patterns. If we have a look at this example of C. elegans embryo, so then we appreciate that, so this is a single cell stage, and C. elegans is a roundworm. At a single cell stage, two types of proteins that are here shown with red and cyan interact with each other in space such that this polarization, okay, it doesn't play again. So the polarization emerges with two distinct domains that predefine the polarization of the embryo, which later coincides with the body axis of an adult worm. So this process is crucial for development of C. elegans. And its mechanism can be formulated as a such reaction network where two species, A and P, mutually inhibit each other such that this complex spatial pattern emerges. There are many other examples where the mechanism of spatial development can be described by reaction network. And this example is also shown here, the digit patterning in mouse. So this work has been done in the lab of James Sharp, who is the head of MBL in Barcelona. And here you can see the time, so the snapshots in time of the developing limb of a mouse with the SOX9 expression pattern on it. Over time, SOX9 gets organized into this spatial pattern, so the striped pattern. And SOX9 predefines the position of the digit of an adult mouse. So, and this mechanism of formation of such a pattern can be captured by the following reaction network. So when we study patterns in developmental biology and we study the mechanism, we don't know yet about how it exactly works, and we formulate our hypothesis in a form of such network before we publish it in Science or anywhere else. First, we need to confirm that indeed this reaction network captures the dynamics that we want. However, so to do so, to validate this, to validate the fact that the hypothesis that is formulated in the form of such reaction network captures the dynamics, we need to first translate it into a spatial temporal model and then to solve it. So when we translate it to a spatial temporal model, it may look like this, but so what we see here is the partial differential equations that describe how the concentration in space and time is driven by the reaction part that purely comes from the reaction network, and we add the diffusion. So this set of partial differential equations are called reaction diffusion models. And however, in order to solve it, such a system, we need to somehow fix the parameters. And because we formulate this interaction as a hypothesis, of course, we don't know the values. So, and these parameters that are reaction rate constants, diffusion constants, total concentrations of proteins, they form so-called parameter space of a model. And it can be quite large. Now, if we have a look at the first system and assume that for example, if we had just two unknown parameters, then in two-dimensional parameter space, we can do extensive screening of the parameter space, right? If you take, for example, 10 points per dimension, then in 2D it's going to be 100 points, in 3D 1,000, while like for low-dimensional parameter spaces, this approach is feasible, and it allows us to explore the whole parameter space and to understand what kind of dynamics this model is capable to perform. However, in high-dimensional parameter space, this approach becomes computationally expensive. So, and here exactly where my framework that is design centering for robust estimation comes into the game. So my framework allows to, first of all, for any kind of reaction network, answer the question whether the model forms a pattern or not. It does it with the computational cost that scales polynomially. So we require less points in the parameter space in order to answer the question whether the model is capable to form a pattern or not. And coming back to this exponential scaling example, if we do the same for the design centering framework, then we can immediately appreciate that for dimensions that are more than 4 already, or like at 8-dimensional parameter space, we need 5 orders of magnitude fewer points to estimate the volumes in the parameter spaces. And also, the framework outputs the design center. So if we have a look at the region in the parameter space where the model is capable to form a pattern, then design center will be its geometric center, so the most robust point. And also the framework outputs the volume of the region of the parameter space, which is nothing but the robustness measure. The larger the volume of the parameter space where the model is capable to form a pattern, the higher the perturbation under which it's still capable to do so. So this framework is massively based on LP adaptation, which is an algorithm that has been developed in our group by a former PhD student. And LP adaptation is a statistic adaptive proposal method for approximate design centering and volume estimation. And here, just for simplicity, I show a two-dimensional example where, so how this algorithm works. So LP adaptation samples uniformly points from LP balls, and here in 2-dimension, L2 ball is just an ellipse. And for every sample point, it evaluates the oracle function. The oracle function is something that we need to define such that this oracle function would give us 1 if the point belongs to the feasible region. So I will call it the feasible region that we want to evaluate, or 0 otherwise. And over time, after sampling from this LP ball the points and evaluating the oracle function, the proposal distribution gets adapted such that it tries to cover the feasible region as much as possible. So at the moment of the adaptation, so during the adaptation, also the heating probability is estimated, which is the probability that a randomly sampled point from this distribution belongs to the feasible region. And in the end, we estimate the volume of the region such, so as the heating probability times the volume of LP ball, which can be computed analytically given its radius and the and the covariance matrix. And the last evaluations are averaged to output the volume of the region that we want to evaluate. So again, here for demonstration purposes, I show a two-dimensional example, but we keep in mind that this algorithm has the highest efficiency in higher dimensional parameter spaces. Now, if instead of this geometric problem, we would use some other or geometric oracle function, we would formulate another oracle function which would be characteristic for the reaction diffusion system, then we can apply it to reaction diffusion models. If you have a look here at the parameter space of a reaction diffusion model, then it would normally, so actually it comes from the PAR system for the polarity establishment. So again, for two-dimensional case, there are 4 regions that are here, and they are bistability region, the instability region, pattern formation region, and pattern maintenance region. And altogether they form so-called characteristic regions in the parameter space for this reaction diffusion model. Now, if we formulate the oracle function such that it captures these features that are characteristic for these regions, then we can use LP adaptation to estimate the volumes. And again, it's more efficient in higher dimensional parameter spaces. Now, how the framework looks like. So we start with the formulation of the oracle function, depending on which region we want to evaluate, and we set the bounds on the parameter space. The bounds can be large, so in our application we used 8 orders of magnitude, which cover the biologically relevant values for all possible parameters. Then we pick a random point from this parameter space and we evaluate the oracle function. So if we are lucky and the firstly picked point belongs to the feasible region, then we right away can start LP adaptation, because as I mentioned earlier, for LP adaptation to start, we need to know the first feasible point. And then we estimate the volume of the feasible region and we are done. However, more often it will happen that we will sample the point that lies outside of the feasible region. In this case, we can't start LP adaptation, but here we came up with a simple and elegant solution, which is just negate the oracle function, which means we just flip the values 0 and 1, so that for the negated oracle function, this point becomes feasible, which means that we can start LP adaptation, we can compute, and we do this until we sample the first point from the feasible region. Once we found this point, we can again go back to the initial definition of the oracle function, or in the other case, if our parameter space doesn't have any region, which also can happen. So quite often it happens that the reaction network just is not capable to give rise to any pattern. Then we can estimate the whole volume and compare it with the volume of the whole parameter space computed analytically and conclude that the model is not capable to form any pattern. So now, having in hand this tool, what can we do? Now I will show you the application of the framework to RAB5 system. So RAB5 is a marker protein on early endosomes. If you have a look at the microscopy image of a cell, so we see here the nucleus, and with green, RAB5 is marked. So if you zoom into these small vesicles, then we right away see that RAB5 doesn't cover homogeneously all the membrane of early endosome, but rather form pattern. And this pattern is thought to be important for the functionality of endosomes. So this pattern has been reconstituted in vitro, which means like just in a tube, right, where instead of endosomes, lipid-coated beads were used, and then all necessary components were added such that such a pattern was reconstructed. And this example gives us the minimal machinery that is needed for such pattern to form. And what is this minimal machinery? So first of all, we know that RAB5 is a membrane protein that is anchored to the membrane, and it can be in its active, functional, or inactive state, depending on whether it's bound to GTP or GDP. It can get hydrolyzed, and there is another component, rabaptin, RABX. If you look at the structure of this protein, that it has this GAF domain that is capable to activate inactive RAB5, and also rabaptin has a binding site for active RAB5. It means that it can recruit from the solution this complex and bring to the proximity to the membrane this activator, which in turn can activate more RAB5, which in turn can recruit more rabaptin RABX, and the positive feedback loop is established. For biologists, this works like a good explanation how the pattern emerges. However, okay, and one more component that I forgot to mention is GDI that ensures the shuttling of inactive RAP5 from the membrane to the solution. So, as I said, for the biologists, this explanation works well, and they think that, okay, there is a positive feedback loop, therefore this model explains the pattern formation. However, when we put all these interactions that I've just mentioned into a network and then use my framework to analyze whether it's capable to form a pattern or not, that we found out that it's not capable to do so. So now, just for the sake of time, I will say that we have introduced 3 additional interactions that we think may happen within, like among the Q components that we put in the tube. And we analyzed these models also with the framework, and it turned out that there are 2 interactions that are important for the pattern to form. And what is also interesting about the volume of the instability region that we've measured, that it compares well, which means that the stability properties of these models come from the basic interactions that are included in this model. However, in order to trigger this capability of this model to form a pattern, we need additional interactions. And now I would like to sum up. So today I have shown to you how, given a reaction network that we think explains or captures the mechanism of pattern formation, how it can be analyzed, and the question whether it's capable to form a pattern or not can be answered. We require less points for screening the parameter space than extensive screening, and which means that the framework becomes more efficient in higher dimensional parameter spaces, and it also puts design center and volume of the pattern forming region, which is nothing but the robustness measure. Also, I've talked about RAP5 system, and I've shown to you that there are 2 interactions that are sufficient to be added to the basic interactions that would explain pattern formation. And also, what is important about this framework is it's very generic, and the fact that we applied it to RAP5 system doesn't really limit it. What is an important output of this framework is that it allows us to formulate biologically meaningful predictions. And by now, I have to mention that these 2 interactions are indeed confirmed that they take place in vitro and in vivo systems. And yeah, and if we are lucky, then we can test them, what exactly happened with our system. And in the end, I would like to acknowledge the whole bunch of people who helped me during this work, and I would like to thank you for your attention. I'm happy to take questions.
Thank you very much, Anastasia. Questions? So I have just one second, very naive one from my ignorance, right? So how do you define a pattern? So the first example that you show from James and the mouse limb development, this is a huge system, multicellular, whatever, but then it seems that the one that you're playing with, I mean, it's kind of clusters of activation of certain pathways within a cell, right? So how do you define a pathway? A pattern.
Yeah, I mean, it depends if we just look at the image and we want to say whether we observe any pattern or not. In principle, even if you look at the noise, right, we always can boost the contrast such that we kind of see a pattern. And here there's the question that how to normalize the concentration such that we know what is the kind of variation in the concentrations that would allow us to tell us what the pattern is. And I think in this case, it depends on the size of the system, because in our institute, there are also groups that work with the so-called stochastic so the patterns that are driven by stochastic noise. In this case, the amplitude of these patterns are really small. We work with deterministic models, so in this case, it's easier to define, so we easily just can set a threshold on the intensities and say if it's larger than a certain value, then it's a pattern.
Okay. Sorry, here.
Maybe I missed it in your talk. Thank you so much. I had a question in the case of dividing cells for the oracle functions. Let's say, because in your system, I didn't recall really well if it was about dividing cells. Let's say you have a dividing cell and one fits well in the oracle function and the other not. Where do you go, and then which path do you take from?
You mean
from
the
there?
patterns that emerge on the cells, or?
how to validate your algorithm when you have like multiple, is it synchronous or asynchronous also? You didn't really tackle this
Yeah, I don't
aspect.
work with the cells, but I work with these vesicles, but I think the approach would be the same, that we get statistics on the distributions of intensities, and then we kind of use these data, like mean value and the standard deviation. Or if we clearly can see that there are different outputs, then we maybe can cluster and deal with these cases separately. Yeah.
I have a question regarding limb formation, specifically limb malformations. So this is work that's done in Stefan Mundlos' lab at Molgan, and I'm wondering if such a model can be used to predict the effect of a genetic change which will cause such a malformation, and maybe even come up with therapeutic strategies, and we can simulate this.
Yeah, actually, of course, like as any kind of model, these models also have their constraints where they're applicable, where not. And I guess if you talk about genetic manipulations, then, I mean, if this gene was included in the model, then I guess it can be used for validation. Or, it's like one way you can use it for validation, or another way you can keep it for like by comparing or checking whether the prediction of the model is correct.
Okay.
So in theory, so if you include these species in the model, then it should give you the correct answer. Otherwise, it has to be changed.
Okay. And one question regarding how long it takes to make such a simulation. Can it be done in a reasonable amount of time? Or?
It depends on the size of the system. So far, we've tried up to 30-dimensional parameter space, and it took around 6 hours to run, but on a single core. This code also can be parallelized, and yeah, then you can run it faster.
Okay, thank you very much, Anastasia, and all the three speakers of the session.
Thank you.
And I hope you found it interesting enough so that you can come back after lunch.